Salamat po sa panibagong araw na ito upang kami ay matuto. Buksan niyo po ang aming isipan upang lubos na maintindihan ang aming mga aralin. Bigyan niyo din po ang aming mga guro, mga magulang at mga kamag-aral ng lakas ng katawan at isipan na masigit namin kailangan sa araw-araw sa panahong ito. Ang lahat po ng ito ay aming dinadalangin sa iyo, aming Diyos Ama. Amen.
My dear students, here are the following reminders during our online discussion. 1. Be on time. Make sure that you log in on your account and follow the given schedule of our discussion. 2. Sit comfortably. Refrain from lying around and instead, sit comfortably and properly. 3. Avoid unnecessary comments on the chat box. Refrain from sending unrelated sticker comments, phrases in our comment section. 4. Stay focused and avoid interruptions. Find a quiet and comfortable place to listen in our discussion. 5. Take down notes while listening or while watching. Make sure that you have a pen and paper with you throughout our discussion. 6. Respect anyone's idea and opinion. If somebody is talking, you need to listen and respect his or her idea. 7. Extend help to your classmates. Let us all learn together and enjoy the rest of our discussion. Good morning, graded learners. Welcome for today's class. By the way, I am Teacher Raymond and I am your science teacher for this class. But before we proceed to our proper discussion this morning, I have here some pictures and I want you to guess what these pictures depicts. Are you ready, students? So here are the following pictures. Flash on your screen. You got it right. It was indeed a microscope. Let's have a short trivia. Did you know, class, that when you shake someone's hand, you also transfer bacteria along with it? In total, you have about 1.8 square meters of skin and there are more than 1.5 trillion bacteria live on it. That's why we always advise and reminded to always wash our hands and keep safe. Because these bacteria are too small to be seen by our naked eye, thus the scientists and us need to use a microscope. So do you have now any idea of what our lesson for today? Very good. Today, we will be talking about the parts and function of a microscope. But before we proceed to our proper discussion this morning, let me present to you first our learning objectives. So at the end of the lesson, you as a learner are expected to first, identify the different parts of the microscope. Second, discuss the function of each part of the microscope. Third, recognize the importance of the microscope in the study of biology. And lastly, familiarize the different techniques in using and manipulating of the compound microscope. So now that you are already know our learning objectives, let's have first a pre-assessment so that I could assess what you already know about our topic for this morning. So students, get your paper and pen and answer the following questions. Flash on your screen. Direction. Choose the letter of the correct answer that corresponds to the question. Number 1. For number 2. And for number 3. number. So how's the result of your test? If you've got a perfect score, well, congratulations, because that means that you have now some idea about the content of our lesson for this morning. While if you've got some mistake, don't worry, because it's just only a pre-assessment. It only used to assess what you already know about the content of our topic for this morning. So are you ready, students? Now let's begin our discussion. So what is a microscope? So microscope is an instrument that used to observe small objects like bacteria, fungi, viruses, and other microorganisms, and even our cells. Microscope also allows you to see things due to its properties such as magnification and resolution. So what is magnification? So magnifying power or magnification is the ability of the microscope to make images or specimen bigger. And the magnification of a specimen can be calculated by multiplying the number found in the eyepiece, which is usually 10 times, with the number found in the objective, which is usually 4 times, 10 times, or 40 times, that is being used. While the resolving power or the resolution, it is the capacity of the microscope to distinguish small gaps between two separate points which humans cannot distinguish. And also the resolving power of a microscope depends on the distance between two separate points or two objects. There are two types of microscope, the simple and the compound microscope. So first we have the simple microscope. So it was named simple microscope because it only composed of one lens and provides relatively low magnifying power, which is 5 times. And one of the good example of a simple microscope is a magnifying glass. And the second type of a microscope is the compound microscope. It is also called as the light microscope. It is composed of 2 or more lenses which provide powerful magnification compared to that simple microscope. It also uses diffuse light, a light which is not harsh and direct from the sun, or artificial light to illuminate the object or the specimen to be observed. The parts of the compound microscope are categorized into 3. So first we have the illuminating parts. These are the parts of a microscope that provides light. So the light may come from 2 sources. First is the natural source, which is reflected sunlight, and next is the artificial source such as the light bulb. So the substage condenser, the iris diaphragm, and the light source are some of the illuminating parts that can be found in the microscope. The second is the magnifying parts. So these are the parts that enlarge the object or the specimen. So this includes the objective lens and the ocular lens. And the third one is the mechanical parts. So this includes the base, the C-shaped arm, and the stage. So these are the parts of the microscope that supports, adjusts, connect, and move the other parts. So now let's talk about the parts and functions of the compound microscope. So first is the eyepiece or the ocular. It is named as eyepiece because it is usually the lens that is closest to the eye when someone looks through the device. So the eyepiece or the ocular lens is located at the top of the body tube where the observer peeps through at the enlarged image of the specimen. So the eyepiece also magnifies the object or the specimen 10 times. Next is the body tube. It is the cylindrical part which connects the lenses of the objectives in the ocular. So the body tube, it is a hollow tube wherein the light travels from the objective to the ocular. It also contains a prism at the base of the tube that bends the light rays so that it can enter the inclined tube. Next, we have the coarse adjustment knob. It is the large knob that moves the body tube up and down to focus the specimen viewed under the low power objectives or the LPO. And also the force adjustment knob is located closest to the arm of the microscope. Next is the fine adjustment knob. It is the smaller knob that is used to get sharp focus of the specimen viewed under the high power objectives or the HPO. And also fine adjustment knob is located further away from the arm of the microscope. Next we have the revolving nose piece. It is a rotating piece located from the bottom of the body tube that holds the different objectives, and it allows the shifting of the objectives from the low power objectives into the higher objectives. And next we have the objective lenses. So this is the lens located under the revolving nose piece which gather light and magnify the image. So there are three types of the objective lens. First, we have the low power objectives or the LPO, which enlarges the specimen 10 times, and it provides the general view of the specimen. And the second one is the higher power objectives or the HPO. It enlarges the specimen 40 times, and it provides the detailed parts of the specimen. And lastly, we have the scanner power objective lens. It only enlarges the specimen 4 times. And the next part of the compound microscope is the stage. It is a platform with an opening to let the light pass wherein the specimen in a glass is mounted. And stages are often equipped with a mechanical device that holds the specimen slide in place and can smoothly translate the slide back and forth and as well as from side to side. Next we have the stage clip. It is the part of the compound microscope that holds the slide in place so that it will not move. And next is the arm. It is the part of a microscope that supports the body tube and the part that is held when the microscope is carried from one place to another. And next is the diaphragm. It is one of the illuminating parts which regulates the amount of light that will pass through the stage. Next we have the mirror. It is the one that regulates the light from the source to the specimen for viewing. And next is the inclination joints. It is the part of a compound microscope that allows you to tilt the microscope into different angles. And the last part of the microscope that we are going to discuss is the base. It is the part that supports the entire body of the microscope. So those are the following parts of a microscope in their function. It's activity time! Get ready, students. Prepare your paper and pen and answer the following activity flash on your screen. The title of our first activity is Label My Parts. What you are going to do is to use the words from the box to identify the parts of the microscope. Then write the matching word on the line which points to the part of the microscope. Now let's continue our discussion. And at this moment, I will be discussing to you the different techniques in using and manipulating a compound microscope. So for the letter A, we will talk about the following techniques on focusing a compound microscope. So when you are focusing a compound microscope, first thing you need to do is to get the microscope from its box or from its cabinet. So you can do this by holding the microscope with both hands. So the one is supporting the base and the other one is holding the arm. Next is put it on a flat tabletop slowly and carefully with its arm facing you. Then place it about 17 centimeters or about 6 inches away from the edge of the table. And next is you should clean all glass surfaces with the use of the lens paper. So you can start this with the eyepiece, then after that, do the objectives, and end with the mirror. So use a clean tissue paper or soft cloth like the old t-shirt to clean the mechanical parts of the microscope. After that, incline or tilt the body, facing light in such a way that you are comfortable with it. Then next is, you must look through the eyepiece or the ocular with one eye, but do not close the other eye. And remember that you always need to keep both eyes open while looking into the microscope to avoid eye strain. After that, you should adjust the mirror so that the light will focus towards the hole of the diaphragm. Caution! You should be reminded, class, that never use a direct sunlight as a light source to view the objects under the microscope, because direct sunlight can burn the retina of the eye and cause blindness. And next thing you need to do is to adjust the diaphragm under the stage so that a medium amount of light gets through. After that, place the slide on the stage. Hold it firmly with the slide clips on each side. Center the specimen on the slide over the stage window. Watching from the side, carefully lower the body tube using the coarse adjustment knob until the end of the lower power objective or the LPO almost touches the cover slip. Then, you must look through the eyepiece with both eyes open. Closing the other eye can strain your eyes. And remember, everything is backward and upside down. Moving the slide to the right puts the image to the left and vice versa. Then slowly turn the coarse adjustment upward and downward until the specimen appears clearly. And lastly, to shift to the high power objectives or the HPO, you need to raise the body tube first using the coarse adjustment knob. And looking from the side, turn the revolving nose piece to the high power objectives or the HPO in place. Use the fine adjustment knob to lower the high power objectives till it almost touches the cover slip. Then look through the eyepiece and adjust the fine adjustment knob until the specimen is seen clearly. Then current microscope models are said to be parfocal. This means that the image in clear focus under the low power objective remains focused after shifting to high power objective. And those are the following techniques on focusing a microscope. Now, let's proceed to the letter B, which is storing a compound microscope. So when storing a compound microscope, first thing you need to do is to turn the revolving nose piece until the low power objectives or the LPO is in place. Next, slowly lower the body tube so that the end of the objective is approximately 1 cm above the stage. After that, position the clips so that they do not exceed beyond the sides of the stage. The next thing you need to do is to rotate the diaphragm until the smallest opening is in position. Then, let the mirror stand on its edge with the concave side facing the user to protect it from the dust. Some of the microscope boxes have a socket for the eyepiece, and in this case, you must remove the eyepiece from the body tube and place it in the socket. After that, put back the microscope's plastic cover, and if the original plastic cover has been lost or destroyed, use any clean plastic bag which is big enough to cover the microscope. And lastly, you should carry the microscope with both hands, supporting the bottom and holding the arm. Then after that, put it back on its box or to its storage cabinet. Next, let's talk about on the following techniques on caring for a compound microscope. So first thing you need to do is to check the microscope before and after use, and report immediately to your teacher when there's any missing or damaged part to your microscope. Next, use a clean tissue paper or soft cloth like old t-shirt to clean the mechanical parts of the microscope. Also, you need to prevent liquids, especially acids and alcohol, from spilling on any part of the microscope. So always use a cover slip in observing wet mount. After that, check for moisture in the eyepiece. This may happen due to the prolonged observation of the specimen. So what you need to do is to wipe it with the use of the lens paper. Also, you must avoid tilting the microscope while observing wet mount because the water might flow into the mechanical parts of the microscope, which causing them to rust. Then you should select a chair with a suitable height so that the both forearms can be rested on the table during the observation. And lastly, you should never store the microscope in a chemical laboratory or any place where there are corrosive fumes. Make sure there are silica get packs inside the microscope boxes or storage cabinet to absorb moisture. Those are the following techniques in using and manipulating the compound microscope. It's activity time! Get ready, students! Prepare your paper and pen and answer the following activity flash on your screen. The title of this activity is Fill Me. What you are going to do is to complete the table by supplying the correct parts, function or functions, and image of a compound microscope. So you need to draw the image of a compound microscope. And after that, the next activity is the analysis question. So this is a group activity. So instruction. Supposedly, you observe the letter E under the low objective, then shifted to the high power objective of the compound microscope. Then you have saw these following images. So for the first image, the letter E appears without the microscope. And for the second, letter E appears under the low power objectives. And for the third one, the letter E appears under the high power objectives. And here are the following analysis questions. And for your assignment, I will give you 2 activities that you need to pass next meeting. And for your first assignment, what you're going to do is to read the selection below and fill in the needed data in order to calculate the magnifying power of a microscope and use the formula ocular times objective. And for your second assignment, review the techniques in focusing a microscope, and what you're going to do is to complete the following by providing the answers from the box below. So I believe that's conclude our topic for today. But before we end our session, anyone who wants to volunteer for our closing prayer? Okay, go ahead. Okay, that's conclude our discussion for this morning. Keep safe everyone, and also don't forget to work with your assignment. Class dismissed. Goodbye everyone.